Hello and welcome to Hello and welcome to Badger Talks Live, which brings exciting happenings, resources, and talent from your UW flagship campus to people of Wisconsin and beyond. I am Brandon Downing, originally from Gary, Indiana. I am a biology major in the College of Letters and Sciences Honors Program, and I am a research fellow on the Chemical Ecosystem Selections in the Origin of Life project. From this research, I met my mentor, friend, and role model, Lena Vincent. I am pleased to introduce Lena Vincent, PhD candidate and a NASA Earth and Space Science and Technology Research Fellow pursuing a special committee PhD in astrobiology and prebiotic chemistry. Today, Lena will be discussing how her research teams work on chemical ecosystem selection and how it may help advance our understanding of how life arose on early Earth and assess the potential of life on other worlds. Lena obtained a BS in molecular cell biology and physiology and a biotechnical certificate from California State University, Long Beach, and an MS in biology from California State University, Northridge. Please welcome my mentor, Lena Vincent.
Hi, everyone. Thank you, Brandon, so much for that really nice introduction. Of course, I'm passionate about my research, but one of the things that I've loved so much about my time here as a PhD student is getting to mentor and get to know students like Brandon. So it's my honor to have Brandon here introducing me. And I'm delighted to welcome you all here to my presentation for Badger Talks Live. I'm going to tell you all about my research on the origin of life and how I'm trying to apply that knowledge in the search for life on other worlds. And before I do that, I just want to encourage you to use the live chat function to enter your questions. There'll be ample time at the end of my presentation for me to answer those questions. So anything you can think of related to my presentation or anything you're dying to know about what we know about the origin of life and the search for life on other planets, please feel free to input those at any time. So I'm going to start, before I dive into my research, I'm going to give you a bit of a larger context about what it is we're doing in our research lab by telling you about the field that I'm a part of. And that field is called astrobiology. And as a relatively up and coming field, and certainly a hot one these days, I figured I would take some time to tell you about what astrobiology is, because I realize not everybody might be familiar with what astrobiology is or the kinds of science that astrobiologists do. So just to kind of give you a flavor for the different kinds of astrobiology science that are possible, in case I have the privilege of being the one to tell you about astrobiology for the first time, and you realize that it's something that you're interested in, I think this will give you kind of a lay of the land with respect to the different kinds of science you can do as an astrobiologist. So I'll start with that. And then, of course, I'll transition over into what our research lab is doing specifically within the astrobiology field. So let's get started. So what is astrobiology? So if you like definitions, the kind of classical definition that you'll see pop up if you do a quick Google search is that astrobiology is the study of the origin, evolution, particularly the early evolution, distribution, and future of life in the universe. And this is kind of the common definition that appears on things like the NASA Astrobiology Institute, which is the largest funder and organizer of astrobiology research in the United States. And I like definitions too, but sometimes it's cool to think about what kinds of questions you get to ask as an astrobiologist. And so let's go through some of those now, because I think personally, some of the questions you ask in astrobiology are some of the grandest and most interesting you can ask as a human being. And I just think it's amazing to think that you can turn these questions into a viable research career. So these questions include things like, what is life? Like really, what is life? What makes it so different from non-living things? What is it all about? How did life get started on this planet? This is just a massive mystery that we have yet to resolve. And also related to that is, can life have originated anywhere else? Does it exist currently on other worlds? And if it does, how do we go about detecting it? And finally, as we continue to expand outward in the solar system, in terms of our exploration missions and even our manned missions in the future, what does that mean for Earth life? So I'm hoping you can see that these questions, as huge as they are in their scope and their philosophical impact, they're also huge in terms of the range of different kinds of science and thinkers that are required to peace and understanding about all of these different things. And so as a result, astrobiology is highly interdisciplinary. That means it merges together lots of different lines of thinking, lots of different approaches that are commonly used in more defined disciplines, things like geology, biology, math, astronomy, and that's kind of nicely represented here in this graphic. But the one thing I'll mention that's not represented in this graphic are the humanities. So as you might imagine, when you're asking questions like, what is the meaning of life? And are we alone in this universe? You start to run into some ethical questions as well. So ethics is a big part of astrobiology, as is philosophy. I mean, can you think of any more existential questions than these? So just thought I would mention that there's a place for the humanities in here as well. Okay, so now that you have a kind of a broad definition of what astrobiology is, I just want to take a step back and acknowledge something very important about astrobiology, and personally what I find super exciting about it, in addition to its highly interdisciplinary nature. And that is that the two biggest mysteries in astrobiology, the two biggest discoveries, have not yet been made. And that makes it a really exciting time to be in astrobiology, because personally, I think we're really close to answering or resolving these mysteries. And that is whether there exists life beyond our planet. So to date, we don't have any conclusive evidence of life that exists beyond our world. There's some question about whether we've succeeded in creating other examples of life here on Earth, either in silico, in computers, or using synthetic biology approaches. But at least we know for sure that we have not found life on another planet beyond Earth. The other big mystery and discovery that's waiting to be made is the mystery that is the origin of life on this planet. We still are clueless, really, really clueless when it comes to how that happened, why it happened. And you might be thinking, well, then what is astrobiology really about if we don't have any kind of answer related to these two discoveries? And I'm here to tell you that there's a lot of optimism in the astrobiology community, and I count myself as an optimist. I think optimism about our ability to answer these two questions is kind of a prerequisite to be an astrobiologist. So that's good news. That means there are people taking this seriously and trying to tackle this scientific problem. But we still haven't made those discoveries. And I think that this is kind of a nice segue into our lab's research and how we're trying to figure out an answer to a related question that we think is at the core of why we haven't been able to resolve those two mysteries of the distribution and origin of life on this planet. And that's because we actually really don't know what life is. And that might come kind of as a surprise, given how much we know about life as we know it around us. But we really don't understand what life is as a general phenomenon, and that's keeping us from making progress on these other fronts. So I think that we all have this intrinsic, maybe even instinctive ability to tell what's alive and what's not. Okay, I'm back. Sorry about that. So as I was saying, we have this kind of intrinsic ability to tell what's alive around us, right? So if we look around a room or even outside, we can kind of point to things and figure out pretty conclusively if something is alive or not. And that might be shaped by what we've learned or what we think is characteristic of life. So things like its cellular organization, the fact that living things tend to be organized in these little bags of chemicals or cells. The fact that life is based on genetics, right? The storing and processing of information that's transferred over time through generations. So we have a pretty good handle on biological characteristics, things that characterize life. What we don't know is whether any of those things are required for the emergence of life, for the origin of life. In other words, we don't know which of these features are just accidents or byproducts of life's evolution, and which of these were actually required for the origin of life, and what might be shared in common with forms of life that might exist beyond this planet. Now, a related problem that's kind of a natural consequence of this lack of understanding is the fact that we don't have a universally accepted definition of life, which makes it difficult to do science with, because although it's great to have different interpretations of what life means and have lots of different circulating definitions of life, in science it gives us a moving target. And it's really difficult to try to study the origins of a phenomenon if you don't really actually know what the phenomenon is. So for now, until we can get more information about what those general characteristics are and how they relate to those essential features, we solve the problem by just being very clear about what we mean by life. And that's exactly what I'm going to do so that you know what I mean when I say life from now on in my presentation. So the definition that we like to use in our laboratory, and that's pretty popular in the origin of life community, is this definition that life is a self-sustained chemical system capable of undergoing Darwinian evolution. And this is a definition that was the result of a NASA working group that was put together almost 3 decades ago now. And they were tasked with trying to come up with a universal definition. And this is what they landed on. And I think it's interesting to point out that the two kind of main features of this definition are this idea of a self-sustained system. And this kind of implies things like self-reproduction, growth, self-replication even. And then the other major component is evolution, right? The ability to generate complexity through an evolutionary process. So this is the definition that we like to stick to in our lab. And I just want to put this out there that this is just one of many circulating definitions in the origin of life community. And not everyone agrees that these are the features that we should be aiming for. But at least in our laboratory, we think that these two processes of self-reproduction and evolution are pretty good candidates for essential features of life. Now, given that we have emphasis on these two processes, I think it's cool to take a step back and think about how this affects the question of how life originated. Because with these two processes in mind, we can reformulate the origin of life problem as wanting to figure out the origin of those two processes. So in other words, what was the first entity to arise that was capable of reproducing itself, so sustaining its own growth in space and time, and becoming more complex through evolution? And so there are lots of different ways and lots of different ideas about what those first entities are. And I'm just going to walk you through them now so you get a pretty good tour of the current state of origins of life research. So one camp is this compartment first camp. So they think that given life's cellular organization, that a likely candidate for the first living system was simpler cells, what are often called protocells. So these are just very simple bags of chemicals that can perform these key life functions. One other camp is this genetics first camp, which imagines that the first systems capable of displaying these life processes were instead genetic pieces of information. So things like RNA, and you may have heard of the RNA world before. And then another camp is this metabolism first camp, which instead argues that the likely candidate for a simple system that can display self-propagation evolution are these metabolic cycles that as a whole comprise the complexity of metabolism of modern life, but in earlier times may have been much simpler. Now, there are, of course, lots of different origin of life theories that fall somewhere in between all of these. And I definitely categorize ours in that category. And so I do want to mention that there are problems with all of these stances. And it's partially because they can't really explain how each of these features, so compartments, genetics, metabolism, how those can emerge spontaneously by themselves on the early Earth in the absence of a prior living process. So that's a major problem. I'm not going to get into any more detail because I just simply don't have time, but just suffice it to say that there are issues with all of these. And what our work is trying to do is amend some of these models or these frameworks to resolve some of those gaps and get us on a path where we can try to explain the origins of these processes without implicating any kind of complicated steps that aren't plausible to imagine arising naturally. So before I get into our specific model that we're exploring in our laboratory, I do just want to dwell on one important question in origins of life research, which is where the stuff you need to make life came from. And you might be tempted to think that this is a big problem and that this is required, you have to fix this problem in order to explain the origin. And thankfully, it turns out you can make a lot of life's building blocks really easily in the absence of life. And we now have lots of different lines of evidence telling us that the early Earth was probably chock full of these building blocks that were arising by lots of different means. And one of those means is spark discharge chemistry. So you may have also heard of this classic Miller-Urey experiment. So that is a spark discharge experiment that was done probably 50 years ago now, more, in which simple gases, things like water vapor, carbon dioxide, methane, nothing sophisticated, was exposed to a spark discharge. And what comes out the other end is many of life's building blocks, specifically amino acids. So this was kind of the first piece of experimental evidence that showed the community that you could make life building blocks really easily in the absence of life. And here's an example of what that setup looks like in a laboratory. We've also since then found that there are other places on the Earth that generate these building blocks naturally. And one example is these hydrothermal vents, which are basically submarine chimney system in which new Earth material that's being produced from Earth's interior interacts with seawater. And the result of that is that these chimneys are basically just giant factories for life's building blocks. And finally, somewhat most elegantly, we've also realized that these building blocks can readily be made in space. And in fact, we have some evidence to show that around the time the solar system was forming, lots of these organics were being formed. So it's possible that the organics that were used by the first living systems actually had these cosmic origins, which I just think is really cool to think about. So the building blocks problem isn't a problem at all. We know that they were probably all over the early Earth accumulating by these different pathways. The big question and the bigger mystery is how you take a sample of those building blocks that arose naturally and get them to self-organize into these complex systems of life. So how did that happen? How do you take these building blocks and get life out of it? Now, I've already shown you that there are different models for this. So maybe the first thing to appear out of these building blocks was a simple bag of those building blocks. They just kind of self-assembled into protocells. They could have assembled into small RNA molecules and given rise to genetics, or they could have formed kind of primitive metabolic networks. But I've already mentioned that there are problems, and so far we haven't been able to piece together a story in which any of these features give rise to life in the absence of a prior living process. And so that's where our labs model comes in. And I've put it here at the bottom because it kind of sums up lots of these different features. So it doesn't fit neatly into any one of those categories. And we call this model our chemical ecosystem model. So let's talk about what that chemical ecosystem model is by first just talking a little bit about metabolism. So I think the chemical ecosystems model can be kind of viewed as an extension of the metabolism first model. So let's go over that a little bit. So metabolism, as you may know, refers to the sum total of reactions that basically make up a cell and help it to maintain its function. So if you sum up all of those reactions, that's what metabolism is. But as you can see from this map of different metabolic pathways, current metabolism is really, really, really complex. In fact, it's almost unimaginably complex. So there's no way that metabolism of this complexity could have arisen by itself. So that's led a lot of researchers to think that maybe specific pathways in metabolism or maybe precursors of those pathways might have been the first thing to arise. And here's one example of the reductive TCA cycle. And people have been able to show that you can get these cycles to arise naturally by themselves in the absence of any kind of complex structures. So it turns out if you just provide metals and some simple inputs, you can get some of these primitive metabolic cycles by themselves, which is really cool. The question is, how do you get evolution? How do you get more complexity out of this, right? Because we're not just looking for metabolic cycles, we're looking for life. So to do that, to explain how we think that these metabolic cycles might represent a path to evolution, I'm going to talk about a process that underlies these metabolic cycles called autocatalysis. So if you remove any of the complexity I just showed you and you stop thinking about the specific molecules that are involved and just look at the net result, that's where things get interesting. So the net result of metabolism is that more of the system is made, right? So metabolism helps that metabolism make more of itself. And that's called autocatalysis in chemistry. And so if we think about how this might manifest in really simple systems, like the one I've drawn here, it becomes pretty clear how this could represent a simple path to these life processes. So in this diagram here, I'm going to use my cursor maybe to point. No, I can't do that. So you can see this cycle between A, B, C, and D, those big letters. So those arrows represent one of those molecules helping the other's synthesis. So if we start from the top, A helps make B using that little a there. And those little letters you can view as food that exists in the environment. So a molecule A, once it's formed, can use food, little a, to make B. B in turn can make C. C can make D. And the key step here is that D can help make 2A. So by the end of this cycle, what you end up with is 2 ways when you started with 1. And you can imagine if you keep flowing that food in, if that food is readily available, these cycles will keep going and they'll grow exponentially because for every 1 A you make 2. Now that's led some researchers to think that as these cycles establish themselves and maybe change over time by adding new reactions, that this could represent a way to generate complexity and maybe even evolution. And so this is just one way to represent that. If we imagine that happening in the cycle I've shown you, it becomes a little bit more clear. So what's not drawn in that cycle is those W's, the waste products. So for every reaction that you see drawn there, there's actually a waste product that usually comes off and doesn't interact with the cycle. It just kind of diffuses away at waste. But there could be rare situations in which that waste product reacts to form another compound, E, which establishes another 4-membered cycle, EHGF. And so this 8-membered cycle now is fundamentally different from that first one, but it required that first one of ABCD to establish itself before it could arise. So our lab has a theory branch that's being led by a postdoc, Zhen Peng, and who's been studying the emergence of these autocatalytic cycles and tried to explain their behavior and how different ecological interactions between those cycles might give us information about the plausibility of these life processes occurring. So here's that autocatalytic cycle I showed you, and I'm going to show you a different way of representing that. So you can basically view each of these cycles as little Pac-Men that eat food that's in the environment, so that little red dot, and that exclude waste, that little gray dot there. And what that side of our lab has found is that when you look at the behavior of these Pac-Men over time, you see this characteristic logistic growth, which is just a fancy term that describes how biological populations behave. So in other words, when you look at how these autocatalytic cycles grow over time, they can actually be described using the same laws, the same rules that we use to characterize fully evolved biological populations, like this pack of penguins here. So this kind of is really cool because it suggests that there's this continuity between how you can describe these simple chemical systems and these fully evolved, fully fledged living systems. And even more interestingly, the theory group has found that when you look at different autocatalytic cycles interacting with each other, so for example, by competing, so maybe 2 of these Pac-Men can feed on the same food and compete with each other in that way, or they can be mutualists. So one of the Pac-Men can produce a waste product that's beneficial for the other one, or you can have one of them just consume the other in true Pac-Man form through predation. When you look at how those behave over time, they also resemble what you see in biological populations. And specifically this predation scenario might look familiar if you've taken an ecology class and looked at how predator and prey dynamics look like, so how those different populations oscillate with respect to each other. It looks the same. So again, just more evidence that there's this continuity between how these ecosystems of autocatalytic cycles interact with each other and how biological populations behave. And so our theory group is now working to figure out how this might lay the groundwork for evolution. So maybe when you have a lot of these different Pac-Men with lots of different kinds of food in the environment and environmental changes and fluctuations, maybe the net result of that starts to look like evolution and you get more complexity in the system. So that's something they're working on now. Another idea I'm going to quickly introduce is what might happen in a real environment. So what I just showed you was simulation, right? So it all happened in a computer. But if we start to overlay this with what we think might have been happening on the early Earth, what does that look like? So a major component of any natural environment, including on the early Earth, was minerals. And that's good because we think minerals played a really important role in the establishment of these autocatalytic cycles. So if you imagine a big ocean with all these food compounds that are very dilute, it's hard to imagine how those could come together and react with each other long enough to establish these cycles. And minerals offer a solution to that because minerals, by their chemical properties, can naturally act as sponges for a lot of these food compounds. So they basically act as little concentrating surfaces where these compounds can stick to each other and interact long enough for these cycles to emerge. So that's how we think minerals play into this. And this is just a diagram here showing how a clay mineral, which you may have heard of before with these sheets, these kind of stacked plates, can stick to lots of different kinds of compounds. And you can see how those compounds, when close together, might be more likely to react with each other. And so we've been thinking about how mineral adsorption, so these systems sticking to minerals, how that might provide a pathway towards evolution. So you can think of these little cores, so that A, B, C, D, and then that E, H, G, F, as different colors, perhaps, stuck to a mineral surface in a real environment like the bottom of an ocean. And we like to call these stuck systems surface limited molecular ensembles, or slimes, and that's what I'm going to call them from now on. So you can view these different slimes stuck to a mineral surface. And the cool thing about a natural environment is that it's very active. So as different mineral surfaces become exposed and old ones get buried down, the net effect is that you might enrich or stabilize systems or slimes that are better able at getting from grain to grain. So the net effect is that you've selected for systems, chemical systems, that are better able at colonizing new mineral surfaces. And this might provide a primitive form of evolution. And so a big part of our lab, and this is work I've mostly been focusing on, is to try to validate that model empirically. So get in the lab and do things experimentally. And we do that by basically just recreating that little seascape I just showed you. So we take vials, we add a food-rich solution, a liquid solution, and mineral grains. And we give time for little slimes to establish themselves. And there might be multiple that come up here as depicted in different colors. And what we do at the end, when we've given time for these systems to establish themselves, is we take a small subset of the grains, usually about 10%, and we transfer them to a new vessel containing fresh food and fresh mineral. And we do that over and over again. And we hope to enrich for those systems that can colonize mineral surfaces faster than the others. So basically they can kind of survive being diluted out of existence through this procedure. And then also a key part of this is routinely sampling both the solution and the mineral grains to figure out what's going on and track whether these slimes have appeared. So I'm just going to give you a quick example of conditions we've tried in the lab. So the really nice thing about this chemical ecosystem selection approach is that it can be used on any set of conditions. So you can use it to test your own hypothesis about the origin of life, depending on what kind of food you think was available, what kind of mineral, things like temperature, the atmospheric gases that were present. So the possibilities are endless, but we had to pick one set of conditions to start with, and we tried to model what we thought was representative of the early Earth as best as possible. So on the food side, so on that liquid side, here's just a quick recipe of what went in there. And you can see there's lots of different stuff, things like amino acids, sugars. And we modeled this off of what you typically find in those spark discharge experiments I told you about earlier. And then I also want to draw your attention to the energy compounds towards the end of the table there. So you'll see things like ATP and ammonium persulfate. So those aren't things we think were necessarily present at the origin of life, but we used them as energy sources to get our experiments started, and then wanted to figure out whether we needed to replace those with something that might be more prebiotically plausible. On the mineral side, we used a mineral called pyrite, which is an iron sulfur mineral, which we ground up into those little grains that we need. And then we also carried out these experiments under anoxic conditions. So one thing we know about the early Earth is there probably wasn't any oxygen at all. So to be faithful to those conditions, we basically use this giant bag that allows us to exclude oxygen and try to represent the early Earth in that way. So we mixed all these compounds together in little vials. So we actually did it in 10 vials just to see if there was replicability. And we basically looked at how these different vials behaved over time as a result of these transfers. And one thing we measured was how much energy the systems were consuming, right? So if systems are growing and evolving, they need energy. So we thought that that might be a good thing to track. And the energy source that we tracked was phosphate. And so you might remember I mentioned that we add ATP in there. So we have a handy test that lets us see how much of that ATP has been consumed by looking at the amount of phosphate that's been released. And when we looked at the amount of phosphate that was released, so that's on the y-axis, across generations on the x-axis for all 10 of our different lineages, so those are different colors on the graph, you can see this cool oscillatory pattern. And just because it might be a little bit easier to see, this is what happens when you average those 10 out, right? So it's the same y and x axis, but here we've just averaged out the 10. And so again, you can see that kind of bouncy oscillatory figure. Now there are lots of different ways to interpret what that means, but for the purposes of this presentation, I'm going to draw you back to earlier results that I'd shown you from the computational and theoretical side, which show that if you had predator-prey type dynamics between these systems, these chemical systems, you could get oscillations. So one interpretation, and of course this needs much more validation, is that what we've managed to enrich in this ecosystem selection experiment is for interacting autocatalytic cycles that have this predator-prey type dynamic. Now, of course, like I said, this needs much more validation, and we're actively working to get more chemical details about the systems that display these oscillatory dynamics to figure out if that's the case. But either way, even if it doesn't turn out to be the case, it just goes to show that chemical ecosystem selection can be used to study chemistry that is consistent with what we view would happen if you had these interacting autocatalytic systems or slimes. Now, another cool thing that we see in those experiments, so this time looking at what happens on the mineral grains instead of the solution, we can take a close look at those using scanning electron microscopy, which allows us very, very high resolution images of those grains. And when we look and compare samples that have a long history of those transfers versus ones that don't have any such history, we see these really beautiful fractal patterns. So that figure on the right, that comes from a sample that had 18 rounds of transfer versus one that had 1 round. And you can see these really cool snowflake shapes on those grains. So we're still trying to figure out what those are. But so far, what we think is that their shape suggests that they're the result of interactions between the mineral and the organics or the food that's in the solution. So again, just goes to show that chemical ecosystem selection yields really interesting chemical patterns that, at least on the face of it, are consistent with what we think would happen if these slimes were to emerge. So here are those fractals there. Okay, so like I said, though, this is all very early work. There's still a lot of work we need to do to figure out if these patterns are consistent with slimes. And remember, to be a slime, you don't just have to self-propagate and arise, right? You also have to be able to evolve. And so that's something we're actively asking right now is, are these slimes evolvable? And this is just hypothetical data here to show you what that might look like, right? So if we have an oscillatory pattern, but what we want to see is a change in the oscillatory pattern over time. And there's different modes that could happen that could happen by. So you could have gradual evolution where the rate of growth increases over time gradually, or you could have this more stepwise or stepwise evolution. So this is just to give you a sense that we know what that should look like. If we start to see a response to selection, in other words, evolution, then this is what we should see. And so we're working on being able to carry out the experiments for much longer and gather more chemical data to figure out if that's the case, both in the system I showed you and other conditions as well. And then on the theory side, they're also working to better understand how evolution could take hold in this chemical realm. By including things like spatial structure, right? So if these systems are stuck to a mineral, then they can start to compete for things like different mineral sites, et cetera. And so they're trying to figure out how this might work to better marry what we're seeing in our experiments with what we're seeing in the theory. So I'm almost out of time, so I'm going to run you through my conclusions. So I hope that I've convinced you that there's still a lot of work that needs to be done in the origin of life field. We still have so much to learn about what makes life special, what those essential features of life are. And I've argued that self-propagation and evolution are two very good candidates for those essential features. And we have a model for how those features could arise in simple chemical systems. And this comes in the form of this chemical ecology framework, which shows how simple interacting systems of autocatalytic cycles can display behaviors that very closely resemble what you see in biological populations and might, in principle, be able to evolve. I've also shown you how experimentally we're trying to validate that model using chemical ecosystem selection, and how, in general, chemical ecosystem selection could be used to screen lots of different conditions and figure out whether evolvable autocatalysis is possible. And then as a bonus, some of the results we've already gotten suggest that this is true. And there's still a lot of work we need to do to validate that. But so far, we've shown that chemical ecosystem selection can produce the kinds of chemical patterns we'd expect to see if evolvable autocatalysis was at play. And I'll just end by bringing it all back out and thinking about how chemical ecosystem selection, by helping us figure out our origins and the origins of key life processes in simple systems, how that might also help us think about the potential for life on other worlds and how we would go about detecting it. So as you know, we have lots of very exciting missions coming up. We just had the Mars Perseverance rover land a couple of weeks ago. There are also other missions like the Dragonfly mission, which will launch in about a decade and look for signs of prebiotic chemistry, so life chemistry on Titan. And so it's really interesting to think about how things like chemical ecosystem selection might be used to figure out if simple life-like behaviors are detectable in these conditions. So we're also working to explore whether chemical ecosystem selection can be used to test conditions representative of Titan or Mars, for example, and figure out if this type of chemistry is possible there as well. So with that, I just want to give a huge acknowledgement to everyone who makes this possible, especially our principal investigator and my advisor, David Baum, the very talented members of our research group, including Brandon, which you heard from at the beginning of this presentation, all of our collaborators, both at UW-Madison and beyond, and then of course, our sources of funding, NASA and the NSF, and you for tuning in. Thank you.
Lena, thank you so much. Hi, everybody. Fran Paleo-Moyer, Badger Talks producer. Wow, you synthesized the origins of life in 35 minutes. So bravo. I'm very impressed. And thank you for sort of simplifying it enough so that, you know, lay people like myself and maybe other people watching can understand it. So very cool work. I'll encourage people to post questions in the chat now. And in the meantime, Lena, I do have a question for you. How did you determine this was the field of study for you? Tell us a little bit about your background and how you ended up studying this.
That's a great question. I love to tell this story. And astrobiologists are a great person to ask that of because they have such nonlinear backgrounds and nonlinear paths to their interest in astrobiology. And that's partially because of how interdisciplinary it is. So my personal background is I actually majored in molecular biology as an undergrad, and I did my research in molecular biology. And then I did a master's in the same area. So I'm a classically trained molecular biologist. And I was tackling very different questions at the time. I was more interested in stem cells and how stem cells influence cancer drug resistance. So very, very different types of questions, but still very interesting nonetheless. And honestly, I discovered astrobiology by accident. I was actually putting together personal statements to apply to grad school at the time. And I was kind of questioning, you know, what am I interested in? You know, beyond the research I'm doing right now, what are the questions that fascinate me? And I remember thinking, you know, oh, I wonder if like, you know, life in space is something that people study. You know, I had no idea. And I did a Google search. I literally remember sitting in that lab at the time, just typing in life in space or something like that. And one of the first things that came up was the NASA Astrobiology Institute website. And the rest is history. I was like blown away to know that this was something that like a real scientist could do and make a career out of and get funding from NASA to do, of all places. I was like, that's it. This is what I want to do. So that's actually what led me to do a master's degree. Is I had this big shift in my interests and I really didn't know what to do about it. I knew I wanted to become an astrobiologist, but it took me a little bit of time to solidify that interest and figure out what I wanted to do. So yeah, so kind of accidental, but it just took me learning that this was a real field of science to kind of toss my hat in.
Going back a little further, were you one of those kids that always won the prizes at the science fairs in middle school and high school?
You know, so that's, you know, no one's ever asked me that. That's a really good question. So I was actually told early on when I was in high school that I could never be a scientist because I wasn't very good at math. So I had a math teacher who, you know, in retrospect wasn't a great mentor just because, you know, I think they were well-meaning and they tried to steer me in a direction that they thought would fit me. And at the time, I was more interested in literature. That was kind of like my strong point. And so they told me, you know, I should aim for that and that, you know, science wasn't really for me. So no, I actually wasn't one of those kids that was very proficient in science or even considered being a scientist because I was told that I would never succeed. But, you know, I happened to be in close contact with people who worked at the Shedd Aquarium. So I lived in Chicago when I was finishing up high school. And I got to do a little brief internship there and fell in love with marine biology. And that's actually what made me major in biology in college, is that little experience there. So it was kind of accidental. And it was it had pure motivations, but no, you wouldn't have been able to guess from my early childhood that this is what I would become.
That's a great message for everybody listening. And tell your kids, folks, like, you don't have to be in science necessarily to become a scientist. So
nope.
I really appreciate you sharing that with our audience. One other question for you. So you're talking about studying the origin of life, but you're doing it within the scope of a single lifetime. Like, can you speak a little bit to that?
Yeah, that's a great question. Yeah, I totally get that. You know, these are things, life was something that took millions of years to evolve on this planet. So what makes us think that we can possibly study that, you know, in a laboratory in a single lifetime, or even in the lifetime of a single PhD? And that's totally valid. And I just want to emphasize that what we're trying to study is those very first steps, right? So the very origin. And right now, given our complete lack of understanding about what that looked like, we don't have a good sense of the time scale, right? We don't know if that's something that took millions of years to take place. We don't know if it's something that took 2 hours. We don't know if it's something that's happening right now, right? So right now, our planet is flooded by life. So if life was originating somewhere, we probably wouldn't know, right? It would probably get eaten up pretty quickly or completely drowned out by life that currently exists. So we really don't know how long it takes for any of these steps to take place. So I think those two things, the fact that we're not really aiming to, you know, summarize the entire evolution of life, we're just focusing on those very early steps. And the fact that we have no idea how long each of those steps took, I think gives us a good impetus for trying to study this in the lab, despite those limitations.
Excellent, thank you. We did have a comment from Harold Steinberg, and he says, excellent speaker, very clear talk on a complex topic. Thank you for taking the time to speak with us today. And I also want to thank you, Lena Vincent, for being with us today and sharing important information with the people of our state. Thanks so much.
Thank you. Thank you. It's my pleasure. I love talking about this stuff, and I'm happy that others found it interesting.
Great. Thanks so much. Please join us next week, Tuesday, March 23rd at noon. We're going to be featuring Anna Simon, who's a reference librarian at the Kohler Art Library. If you're not familiar with this library, it's a hidden gem on campus. She's going to be talking about its history, how and why people research art there, and then she'll show some colorful examples. So please tune in next Tuesday. As always, visit badgertalks.wis.edu, where you can see our upcoming schedule of talks and sign up for our email list so you're the first to learn about new talks being scheduled. Feel free to consider making a donation. Badger Talks operations are supported by a grant from the Alumni Association, so we appreciate those donations to continue free programming. You can also sign up to host a speaker for your own event at that website, so please do visit. Thanks for tuning in, as always, and we'll look forward to seeing you next week. Thanks, everybody.